is drug action? Drug action. Drug action is that it is the what kind of action the body is having, and the drug effect is that what kind of effect the drug is making on the body. Drug action can be agonist or antagonistic, and drug effect can be, it is the ultimate change in the physiology that is happening in your body. So drug action, drug effect. Next it is regarding the principles of drug action. So it is going to be regarding the principles of drug action. Drugs do not impart new functions to any system or organ or cell. They only alter the pace of ongoing activity. So never think that if you're going to take a drug, it is going to give you some extra effect. So whatever effect, whatever functions that is happening in your body, it is either going to stimulate the particular function, depress a particular function, irritate a particular function, replace or make a cytotoxic action. But it is never going to give you some additional effect or some additional function to your body. Okay, it will only confine its actions to stimulation, depression, irritation, replacement, or cytotoxicity. So we'll see what stimulation is first. So it is selective enhancement of the level of activity of specialized cells. Okay, these are the things which comes under clinical pharmacology. So what happens is that in clinical pharmacology, you will be giving the medicines. After experimental pharmacology, you felt that safety, efficacy, everything is there. What you will give? You give this particular medicine to healthy volunteers. And then what will you do? You will actually make relevant tests, relevant the investigations to know the action, and you do program screening in order to know the pathway of this particular drug. This is happening in clinical pharmacology. So what are the changes that is happening in the body? That is exactly what we are studying in clinical pharmacology. So it can cause stimulation. What is stimulation? Stimulation is selective enhancement of the level of activity of the specialized cells. Okay, for a specialized cells, selective enhancement of the level of activity is known as stimulation. So adrenaline stimulates heart. This is stimulation. That is, selectively it is going to activate some property of the body. Excessive stimulation is often followed by depression of that function. That is, if you are going to take a medicine to increase the T3 hormone in your body. So excessively T3 is being getting produced in your body. What will happen? There is a counter phenomenon that is happening in your body, isn't it? When T3 gets increased, TSH will be naturally increased in order to suppress the excessive T3. So what happens when if excessive stimulation is going to happen, it will be definitely it will lead to depression of the function, because the other counterpart of your body will excessively activate so that it will very severely try to control or debar this particular effect. So do not excessively stimulate any of your organs or excessively stimulate any of your hormones or enzymes, because it can in the long term, in the long run, it can lead to the depression of that particular enzyme, hormones, or particular functions also. High dose of picrotoxin, a CNS stimulant, produces convulsions, coma, and respiratory depression. Next, depression or diminution. So this is selective diminution of the activity of the specialized cells. You are going to give a particular drug in order to make a depression of a particular thing, and that is barbiturate depresses CNS activity. Most drugs cannot be called as stimulants or depressants because drugs stimulate one type of cells but can depress the other. So actually, dividing them into depressants and stimulants is actually relative. Okay, relative to the organs where it is being active. Acetylcholine stimulates intestinal smooth muscles but depress SA node in heart. So it means no, relatively, when you say acetylcholine, is it a stimulator or a depressant? You can say that it is a stimulator for intestinal smooth muscle, but it is a depressor for SA node in heart. So this is relating to stimulation and depression. Next, irritation. So what irritation is? Irritation is often the noxious effect and is particularly applied to less specialized cells. Okay, it is epithelium, connective tissue, etc., which is very less specialized. So this is often it is actually a noxious effect. Okay, so where do we use this irritation? Usually irritations will always cause inflammation, corrosion, necrosis, morphological damage, etc. But this kind of irritation is useful in case of respiratory conditions when you want to expel out the mucus inside the alveoli or inside your bronchus. In such cases, irritations are being used clinically. But usually, if a drug is an irritant, then what happens is that continuous usage can cause inflammation, corrosion, necrosis, or morphological damage to a specified cells or tissue. Usually, because it is more or less irritations usually occur in less specialized cells or like epithelium connective tissues, which is very prone to injuries, various kinds of injuries. So, it is not very much advised to use medicines which have an irritating action. Okay, and this can also result in diminution or loss of function of that particular area also, because excessive inflammation, necrosis, or morphological damage can always lead to loss of function of that particular area. This is in regarding to irritation. Next, coming to replacement. Sorry. Okay, replacement. So when it comes to replacement, what happens is that this refers to the use of natural metabolites, hormones, or their congeners in deficiency states. Okay, so here what happens is that you have an insulin deficiency in your body. So you are going to replace the deficiency with a drug. That is which is occurring in replacement therapy when you are using a particular drug. Example, the commonest and the best example that you can give is insulin, which is being given in an insulin-dependent diabetes. That is, you know that there is a deficiency of a particular, you are using thyroxine tablets for thyroid TSH hormones. That these are all replacement therapy. The best areas where we use replacement drugs is in hormone relation only, and we rightly call it hormonal replacement therapy for that particular usage also. You find that a particular hormone is not working or it is diminished in your body, so you replace it with the corresponding measurement of the hormones externally, that is replacement drugs. Next, cytotoxic effect that can cause in the body due to drug action. Selective cytotoxic action on invading parasite or cancer cells, attenuating them without significantly affecting the host cells, is utilized for cure or palliation of infectious and neoplasms. Okay, so that is usually the cytotoxic effects in drugs are usually used in cases where you want to treat a carcinoma. You want to selectively kill a particular set of cells. You want to selectively kill specific neoplasms. So in such cases, in such conditions, what happens is that it is the cytotoxic effect of the drug that we usually use in such scenario. Examples, penicillin, chloroquine, etc. So these are the basis of drug action. So how a drug is acting in your body. So how all can the drug act in your body? Drugs will never give you an extra effect, or it will never cause an extra action. Drugs always act, either it stimulates the function that is happening in your body, or it will diminish the functions, or it will irritate your body functions, or it will replace the functions, or it can cause cytotoxic functions selectively when in cases such as cancer therapies, neoplasms, etc. So these are the types of drug actions. Now coming to mechanisms of the drug effect. We know that pharmacodynamics is about drug action and drug effect. So we already saw about drug action. Now we see about drug effect. Few drugs act by virtue of their property. That is, when bulk laxatives, you are asking a patient to take a lot of fibrous content food, or you are asking them to take a lot of oats, then they will be acting as a bulk laxative because of its physical mass, not because of it is not never going to bind in the receptor, and that head block and all is not going to happen. It is acting because of its physical mass. Okay, next, petroleum jelly. It, because it's of its physical form and opacity, it will be acting on your body. Magnesium sulfate acts on the basis of its osmotic activity. So these are drugs which act on their virtue of their property, and they do not interfere in your physiological functions or anatomical functions. So they act on their own properties. Effects by virtue of interacting with a discrete target biomolecule, which usually is a protein. Usual proteins that are targets of drug action are enzymes, ion channels, transporters, receptors. Okay, and there are few other kinds of, there are very few drugs which acts in this way by its property. All others, it will show the effect based on its interaction with the receptor. Okay, and the receptor can be an enzyme, it can be an ion channel, it can be a transporter or other cell receptors. Next, combined effects of drugs. That is, when two or more drugs are being given for together, then how can they act? So they can be act synergistically. That is, it can either cause additive effect or supraadditive effect. That is, synergism is more. Antagonistic effect. That is, it can diminish. One can decrease action of the other. That is antagonism. It can be physical antagonism, chemical antagonism, physiological or functional antagonism, receptor antagonism. See, these are the four types of antagonism that can happen because of combined effects of the drug. So, if, when you are going to give two or more drugs together, it can either have a synergistic activity or an antagonistic activity. In synergistic activity, it can have an additive effect or a supraadditive effect. Antagonistic activity, it can cause physical antagonism, chemical antagonism, physiological or functional antagonism, or receptor antagonism. So we'll see in detail what each of the subdivisions are. Now, what is synergism? When the action of one drug is facilitated or increased by the action of the other drug, they are said to be synergistic. That is, when the action of one particular drug is facilitated or increased by the action of the other, they are said to be synergistic. Next, it is additive. So what is synergistic? Two types of synergism is that additive action, supraadditive. In addition, A plus B is equal to the effect of A plus B. 1 plus 1 is equal to 2. That is additive action. Aspirin and paracetamol, when you are going to give it together, it is act as both analgesic as well as antipyretic. Supraadditive action, that is 1 plus 1, you are going to get 3 or 4. Then it is going to, drug A plus drug B is going to have an effect more than effect of A plus B. Then it is known as supraadditive effect. The example is acetylcholine. When you combine it with physiostigmine, inhibition of the breakdown would be there. So it is going to have an exceeding action rather than the combined effect of both of them together. That is supraadditive effect. So this is the two types of synergism that can happen when you are going to give two or three drugs together, or more than one drug together, can either cause additive action or supraadditive action when it comes to the purview of synergism. Okay. Next, we see what antagonism is. Antagonism is that when one drug decreases the action of another drug, it is known as antagonism. Okay, they are working contradictorily. The types of antagonisms are physical antagonism, chemical antagonism, physiological or functional antagonism, or receptor antagonism. In physical antagonism, what happens is that based on the physical properties of the drug, that is, charcoal prevents the absorption of poison. This is physical antagonism. So in a poison afflicted person, if you're going to give charcoal, charcoal is going to inhibit the action of the poison which has got inside. That is physical antagonism. Chemical antagonism, they are going to react chemically and form an inactive product. This is chemical antagonism. That is, it is not, it is, they are going to chemically react together and they form a unabsorbable form. They turn into an unabsorbable form. That is chemical antagonism. Next, it is physiological or functional antagonism. When two drugs act on different receptors and have physiological effects in opposite directions. For example, histamine and adrenaline on bronchial muscles. Okay, histamine will go irritate, adrenaline will go relax. This is physiological or functional antagonism. Receptor antagonism. One drug blocks the receptor action of the other. So this will be it will be more or less similar. There is drug and the heads of two drugs would be very similar to others. So what happens is that instead of the drug going and binding to the particular receptor, the other similarly structurally similar other one will go and bind to it, thus causing receptor antagonism. This is regarding to antagonism. So this is in regarding to pharmacodynamics. So what is in nutshell pharmacodynamics? Pharmacodynamics is how the drug is going to act on your body, how the drug is acting on your body, and what kind of effects has happened in your body because of taking a drug. It can be it can be a synergistic action. It can be an antagonistic action. It can cause the replacement action, irritation action. It can diminute. It can stimulate. It can cause cytotoxicity. So these are different types of pharmacodynamics of a particular drug that can happen in your body. So that is about pharmacodynamics. Next is pharmacokinetics. Okay. Regarding the pharmacokinetics, broadly, it is the ADM, absorption, distribution, metabolism, excretion of the drug. So what the drug is doing in our body, what the body does to the drug. So pharmacodynamics is what the drug does to our body. What is the effect that the particular drug is giving in our body. Now pharmacokinetics is what the body does to the body. How does the body absorb the particular drug? How does the body distribute this particular drug? How the body metabolize this particular drug? And how the body excrete this particular drug? Whether it is going to be excreted through urine, whether you are going to excrete it through your feces, whether you are going to excrete it through your sweat, or whether this is going to cause some cumulative action in your body, that is regarding pharmacokinetics. Next, it is absorption. Absorption is the transfer of a drug from site of administration to the bloodstream. This movement of drug from one side of the biological barrier to the other is called as biotransfer. So what is absorption? It is the transfer of the drug from the site of administration to how a particular, when you are going to make it topically, when you're going to take it orally, or when you're going to take it IV, IM. So how does this particular drug which is being given to the body get transferred from the site of administration to the bloodstream? That is regarding to absorption. This movement of drug from one side of the biological barrier to the other side is called as biotransport. And the important transport mechanisms that is adopted for this absorption or this transport is passive diffusion, carrier-mediated transport, pinocytosis or phagocytosis, and filtration. These are the four transport mechanisms that happens in absorption of a particular drug. It can be passive diffusion, it can be carrier-mediated transport, pinocytosis and phagocytosis, or filtration. And carrier-mediated transport can be either facilitated diffusion or an active transport. Okay, so carrier-mediated, either it gets binded to some carriers and getting inside, or it can be an active transport. So these are the important transport mechanisms of the drug. This is a pictorial depiction of passive diffusion. That is, from higher concentration to lower concentration, naturally the molecules will move. Okay. So lipid soluble, non-lipid soluble, aqueous pore or paracellular space. So what happens is that through these spaces, intermolecular spaces, the aqueous thing will come inside. But because this membrane, you all know that the cell membranes we have already studied while we studied about Sneha Vana or Sneha Kalpana, we studied that our cell membranes are lipid soluble. So these lipid soluble things would definitely come out. Okay, that it will get dissolved in the particular membrane, it will come inside. But the aqueous molecules, it will be transmitted to the other area only through the junctions. Okay, membrane junctions, cell junctions only will come. Next, it is carrier-mediated transport. Carrier-mediated transport happens usually, carriers are usually proteins present on the membrane system. So what happens is that a drug substrate plus carrier, they form a compound. And this carrier will carry the drug across the cell membrane. And it can happen at either facilitated diffusion or active transport. So just say photo. See, lipid soluble solutes are they are getting they get binded to the here the carrier things, and they'll allow it to come across. See, it gets binded, it will open up here, it will come across here. So this is how the carrier mediated transport happens. It can be either facilitated diffusion or it can be an active transport using energy. This is cytosis. Cytosis is phagocytosis, pinocytosis. Two types of cytosis are there. It can be either cell eating, phagocytosis, or pinocytosis, cell drinking. Okay, when cells are taking large particles like this, when you're taking large particles, making into phagosome and it is evacuating into the intracellular matrix, that is known as phagocytosis. When it takes in liquid things, Okay, fluids, it takes it and forms a vesicle, then it is pinocytosis. So this is by cytosis also it comes out. Next, movement of the molecules through membranes from regions of high hydrostatic pressure to regions of low hydrostatic pressure. Separation of solids from fluids generally applies only to capillary walls. So filtration is actually, usually it appears in capillary walls. So movement of the molecules through membranes from higher hydrostatic pressure to lower hydrostatic pressure. So that is, it will separate the solids and fluids each other, and usually this happens during the in areas of capillary walls. Filtration happens. Now coming to, so these were the types of cell transport. So it was through passive diffusion, carrier mediated, filtration, or by cytosis. So these are the four methods of cell transport. Next, regarding the routes of absorption. So drug administered can be directly to the blood. Then other routes are absorption via GI tract when you take it something orally, absorption via parenteral route when you give it rectally, absorption via lungs when you're going to take it as vapors or like sprays and all, absorption via topical sites when you're going to apply it on your body, external body. So these are the areas of routes of absorption. Now, how can we delay absorption? Methods of delaying absorption. So using different dosage forms. Okay, so actually, gaseous dosage forms are more easily absorbable than solid dosage forms. So different dosage forms have different types of absorption paces. So by using different dosage forms, it can delay the absorption or it can initiate absorption. Changing the physical characteristics of the drug and adding a vasoconstrictor drug or applying a tourniquet. Okay, if you're going to add on a vasoconstrictor to the along with the drug, along with or you apply a tourniquet, then the blood supply would be lessened. Only when blood supply to an area comes in, absorption will be increased or absorption gets initiated. So when you are going to add a vasoconstrictor to the to the drug you are going to take, or if you're going to apply a tourniquet, naturally blood supply to that area will be hindered or it will be decreased, and that can delay the absorption. So this is different methods of delaying absorption. Methods of speeding absorption. By adding hyaluronidase to injection fluid increases the rate of diffusion through the intestinal spaces and it speeds up the drug absorption, increasing the local blood flow. Applying hot fermentation or massage. This is exactly why we give Snehana and Svedana to our body. This is to increase the, when you have inflammations and all, giving Svedana will increase the blood flow to that particular area. This will help in the reducing the inflammation. So this is methods of speeding absorption. Next, bioavailability. Extent of active concentration of the drug available at the research site of action is known as bioavailability. In other words, biologically available concentration of the medicine at the site of action is known as bioavailability. So in what all does the bioavailability depend on? How good or how, to what extent does the active concentration of the drug gets availed in your body or get availed in at the site of action? So that depends on the pharmaceutical factor and pharmacological factor also. Okay, that is on what kind of drug. Pharmaceutical factors are the particle size, how you are going to put it, whether you're going to give it as powders, then whether you are going to give it as liquids, whether you're going to make it into gel, whether you're going to make it into solid forms. So the particle size interferes with the biological activity or bioavailability. Salt form, whether it is in a salt form, whether it is in a crystal form, whether it has some hydration associated with it, the nature of excipients and adjuvants, the degree of ionization. So all these factors will affect the these are the pharmaceutical factors involved with the drug in order which determine the bioavailability. Pharmacological factors, gastric emptying and gastrointestinal motility. Gastrointestinal diseases, food and other substances. If food or other substances are there, then it will be delayed. The absorption will be definitely delayed. First pass effect, drug-drug interaction. If this drug is going to interact with some other drug, then again, naturally, it gets delayed. Pharmacogenetic factors and miscellaneous. So these all are the pharmacological factors related to the absorption of a particular drug. Next, so this was regarding absorption. Next, we are going to see what is distribution. So distribution means the pattern of scatter of the specified amount of drug among the various locations within the body, and it is called as drug disposition also. Physiological barriers restrict the drug distribution. Example, blood-brain barrier, placental barrier. So these all restrict the distribution of a particular drug. So how this drug is going to get distributed all over the body is meant by distribution. Next, it is metabolism. Drug metabolism usually occurs in the liver, but occasionally it can also take place in gastrointestinal tracts, lungs, kidneys, skin, or plasma. Actually, the biotransformation is enzyme catalyzed chemical transformation of the drugs within the living organism. So, how a particular drug gets transformed inside the living organisms to biologically active form, that is known as metabolism, and it is also known as biotransformation. That is how the particular drug is going to get transformed into biologically active form in the body, in the living organism, that is known as metabolism. Lipophilic drugs will not be excreted. Transformation to hydrophilic helps in excretion through kidney. So usually lipophilic drugs only gets absorbed, but such drugs is very difficult to get eliminated. So what happens that this will be transformed into what is hydrophilic excretors or hydrophilic compounds, and that would be excreted through the kidneys. This needs to be done since drugs and chemicals are foreign substances in our body. So metabolism and converting into a form which can be excreted from the body is very, very important, because it can, otherwise it will cause cumulative effects in your body, isn't it? And it can cause cumulative toxicity also. So this is the relevance of metabolism of a particular drug. First pass mechanism. First pass metabolism is the mechanism by which a particular drug trespasses the kidney and then reaches into the blood. Because it trespasses the liver and reaches into the blood. Because we all know that liver is a filtering organism, isn't it? We all know liver, it is a purification organ, and this liver can purify the bloodstream. So the things which we get, the oral things which we insert, it comes to the GI tract here. See, from the GI tract, the blood will take the drug first into the liver, and there it gets purified. It will sort out the unwanted things here itself, and the wanted things are just given into the bloodstream, where it gets distributed all over the body. So this is the first pass mechanism, where the drugs which we taken passes the liver and then gets into the bloodstream, which will be distributed into a body. See, drug metabolism occurs in liver before the drug enters the systemic circulation. Significant amount of drug is inactivated, so that the results in decreased bioavailability or diminished therapeutic response. So when you know that you're giving a drug which can cause a toxicity effect, then the drug has to undergo first pass mechanism. But when you want to give a drug and you need to get the effect, rapid effect is needed, you want to lower the temperature suddenly, then what will you do? You will exclude first pass mechanism, and you will directly give it as IV into the bloodstream. Something which you give as IV will never pass the first pass mechanism, but something which you take inside through your oral mucosa will definitely enter the first pass mechanism. Next, excretion. Some drugs are excreted out unchanged, but some are excreted out as metabolized in urine or bile. Major routes of excretion is renal, biliary, fecal, and alveolar. Minor routes are through milk, skin, hair, sweat, and saliva. So, accordingly, the things get excreted via these routes. So, this is regarding the pharmacokinetics. So, we have seen the absorption of the drug, the things which can hinder the absorption, speed up the absorption, distribution, metabolism and relevance of metabolism, and also the excretion, different routes of excretion of the drug. Now, animal ethics, that is also an important part, because experimental pharmacology is one part which is important in today's life. So, we have to understand some animal ethics also. So, animals are using research or experimentation in place of humans ethics for various reasons. Because animals are used most often for disease treatment, prevention, treatment of injuries, basic medical testing, medical diagnosis, vaccination, anesthesia, antibiotic prevention. A lot of areas we use animals for experimental pharmacology. So, when we are doing animal studies, we widely use them to establish the safety and efficacy of a new drug. Okay, and these experiments can definitely cause pain to the animals, and it can also reduce the quality of life if they are going to live further, or maybe we are going to kill them also. So it is morally wrong to cause animals such sufferings with these experiments. So most of the times the diseases which we are going to induce are going to make severe complications in such animals. And at the time when they are, most of the time the animals will be sacrificed for histopathology studies also. So all these things are going to harm the animal species a lot. Okay, so we have introduced certain ethics, which is known as animal ethics, to prevent the over torturing of animals. Okay, as usual, there are two school of thoughts for animal ethics: concerned with animal rights and concerned with animal ethics. So, animal rights are that animals can be used to save humans. The group supporting animal ethics have postulated a theory to generate alternate methods of practice. Okay, and conflict is that led to the development of ethical principles of animal use. How animals should be managed and treated by European experimental pharmacology, that comes under animal ethics. They have five freedoms. Okay, freedom from hunger and thirst. You should never make them hungry or thirsty. Freedom from discomfort. You should not like to cause too much discomfort. Freedom from pain, injury, and disease. Freedom to express normal behavior. Freedom from fear and distress. Okay, these are the five freedoms that you have to make sure when you are going to do an experimental pharmacology with any of the animal species. So freedom from hunger or thirst by ready access to fresh water, diet, maintain full health and vigor. Okay, throughout your study. Freedom from discomfort by providing an appropriate environment, including shelter and comfortable resting area. So they have to be maintained at a specific temperature. They have to be given good food. Okay, so everything should be maintained for these animals. Freedom from pain, injury, or disease by prevention or rapid diagnosis and treatment. So when you are going to test an analgesic activity of a particular drug, you should make sure that you are doing the experiments very fast. You should not cause trivial pain and you should not make them suffer a lot. Okay, you find out a particular, you induce a particular disease that you found out, and you have to give immediate relief also with counter treatment. So you should not cause much problems or much pain or injury or disease to the animals for a longer period. It's freedom to express most normal behavior by providing sufficient space, proper facilities, and company of the animal's own kind. Okay, it should not be put it alone in a place or something. Give it the freedom to express its normal behavior. Freedom from fear and distress by ensuring conditions and treatment which avoid mental suffering. So this has also to be considered. These are the five freedoms. Next, alternatives. If you can replace, do a replacement. If you can reduce the number of specimen that has to be taken, try to reduce it. Refinement. If you could refine the protocols, try to refine it also. So what is replacement? Replacement means replacing higher animals with lower animals. Microorganisms, plants, eggs, reptiles, amphibians, invertebrates may be used in some studies to replace a warm-blooded animal. Alternately, live animals may be replaced with non-animal models such as dummies for an introduction to dissection for teaching the structure of animals of human body. Mechanical or computer models, as we have told in silico studies, that can be used as a replacement. Replacing experiments on animals with alternative techniques such as experimenting on cell cultures, using computer models, studying human volunteers, using epidemiological studies, and complete replacement. There is also two types: complete replacement and incomplete replacement. Some part of the study you can do it in animals, some part otherwise. Complete replacement, you are totally avoiding the animals. This is replacement. Next, reduction means minimizing the number of animals, modifying the experimental design to minimize the overall animal usage. So take the only in the confined number, reducing the number of animals by improving experimental techniques, improving techniques of data analysis, sharing information with other researchers. These are reduction. Refinement. Refining experimental protocols to minimize pain or distress. Examples of refinement include identifying pain and distress and making plans for preventing or relieving it. receiving adequate training prior to performing a procedure, because animals and all, they are living organisms. So before going for an animal study, try to get receive a adequate training in regard to animal handling before doing it, using proper handling techniques for animals. Okay, so these are the refinement methods also. So these are the three R's, that is replacement, reduction, and refinement. These has to be adopted while considering animal ethics. So these are in regarding to animal ethics. So we have looked on the five freedoms of animals, and also the three R's, the three replacements or three things by which you can avoid the over usage of animals. Okay, there is reduction, refinement, and replacement. So these are in regarding to animal ethics also. And also refining the experiment or the way the animals are cared for so as to reduce their suffering, using less invasive techniques, better medical care, better living conditions, ensuring correct animal for the type of study, ensuring the drug doses methods, binding up the procedures if the required data is obtained earlier than expected. So these all has to be followed in relation to the animal ethics, because if you are if you could especially we have already talked before also, the correct animal health, if you want to know the teratogenicity, try going for the mice itself rather than rat, or rather than the guinea pigs or something. So choose the correct animal for the relevant type of study, and also the doses etc has to be very priorly planned. And if you are going to get results, enough sufficient data is obtained before the completion of study, make sure you stop the study, because if you are going to again do the study, you are again going to harm many more animals also. If you are going to, if you got enough data, then try to stop it there. So these are the methods by which you could reduce the animal uh, animal harm, and these are the protocols of animal ethics also. So today, by these things we have seen regarding the preclinical, it was various aspects of doing research. Okay, how you can practically apply Rasa Shastra and Panchashtra Kalpana in the context of research. So how we are going to develop a drug. So first you will go for a preclinical study, then you will go for an experimental pharmacology, you then go for a clinical pharmacology, then you give the clinical trials, then post-marketing surveillance. And we have very detailedly seen how we are going to do this drug development. This is a point for your G first year syllabus, and briefly explain drug development procedure. That can be a question. Briefly explain the various clinical trials involved in clinical pharmacology. Explain briefly experimental pharmacology. What are the different types of preclinical trials, and what are the different screening methods? These are all very relevantly and very continuously, repeatedly asked questions in PG first year syllabus of R&D. So make sure in this you will also study in research also. You have very detailedly you will be studying in research, because toxicity studies and all I haven't studied, I haven't mentioned here much deeply, because you will definitely study them in your research. And you have subacute study, acute study, etc., etc. That has to be dealt in very detailed manner. So make sure you go through that also. So with this, we can wind up today's class. Thank you.
उनके पास पैसा और बस छोकरी नहीं है, वहाँ की तो भी सब कुछ है।